Welcome to the Chemistry, Biology and Math Revision Hub. Today we're doing the A-Level Biology topics. This is muscles, movement and the heart. We're beginning with topic 7b1, tissues of the skeletal system. The tissues of the skeletal system include cartilages, tendons as well as ligaments. We can see the cartilage here. These are hard but flexible tissues. They are made up of chondrocytes with varying amounts of collagen fibrils. Cartilage is elastic and it can return to its original size when stretched. And it's a very good shock absorber, we can see here within the joints. The next one is the tendon. We can see this connects a muscle to a bone. It's made up of bundles of collagen fibers. These form relatively strong inelastic tissues. They're entirely made up of white fibrous tissue. And then the ligaments which join bones to bone. These are elastic and they enable bones in a joint to move. We can see this bone is connected to that bone using this ligament. So the ability or motion that occurs around a joint can be ensured with the presence of the ligament. They're made up of yellow elastic tissue which gives them good strength as well as elasticity. Looking at how joints, muscles and bones are involved in movement, the tissues of the skeletal system have to work together to hold the bones. This gives the required range of motion to be attained. They also protect bones from wearing away. Mobile joints contain synovial fluid. We can see around these mobile joints, and this reduces friction. Joints also contain cartilage that ensures smooth movement at the joints. The muscles holding the bones at a joint include extensors as well as flexors. These are going to be antagonistic muscles. Flexors do flex to bend a joint, while extensors cause the extension of the joint. Going on to topic 7b2. What is a muscle? Muscles are made up of protein and they consist of large numbers of long cells. These are called muscle fibers. The muscle fibers are held together by connective tissue. Muscle fibers do contract to shorten in order to do work. And when muscle fibers are relaxed, they are pulled back to their original length. They have good supply of blood to deliver oxygen as well as nutrients for the contraction process. And they can be stimulated by the nervous system as well as stimulation by hormones. The main types include the skeletal muscle. The skeletal muscle is voluntary. It is striated, meaning it has stripes like we can see them here, those stripes, so it's striated. It contracts quickly. This ensures that some activity can be attained within a shorter period of time, for example, running away from something scary or trying to protect yourself from danger. They also fatigue quickly, meaning you cannot continuously make the muscles to be in a contracted state for a longer period of time because fatigue is going to occur. The next one is smooth muscles. These are those found mainly in some internal organs, like we can see the digestive system and so on. These are involuntary, meaning we may not involve conscious decision to make these muscles to work. They are not striated. We do not see any stripes in them. And they contract slowly, they fatigue slowly, and they are found covering internal organs as well as around the gut. The next one is the cardiac muscle. We can see it's cardiac because it's muscle of the heart. This is involuntary. It is striated. We can see the stripes. It has fibers joined by special cross sections. We can see those fibers here joining the cross sections. And then it contracts spontaneously, meaning there is no need of being stimulated by the nervous system. And the cardiac muscle does not fatigue, meaning the cardiac muscle works for the lifetime of a specific organism. Now here we're looking at skeletal muscle and the sarcomere. Muscles, as we can see here, these are composed of long fibrous cells called muscle fibers. They also contain thousands of smaller units of muscle contraction called sarcomeres. We can see this is one sarcomere, or we can see that bigger image, that is the sarcomere. So meaning a single muscle fiber can have multiple sarcomeres, like we can see this is one sarcomere, we see the next one, and then the next one, and so on. The sarcomeres are arranged in a series, forming a repeating pattern along the length of the myofibril. Each sarcomere, like we can see here, is delineated by a Z line. We can see this Z line. There is from the Z disc to the next Z disc. The Z lines composed of interlacing thin and thick filaments, mainly myosin and actin. We can see this one here is myosin and that is actin. So basically in shortening the sarcomere, the actin filaments are going to be brought closer or pulled closer to each other so that the sarcomere can be shortened. Moving on to topic 7b3, the different types of muscle fibers. Here we're going to be looking at slow twitch and fast twitch muscle fibers. But before we do that, let's begin by looking or comparing myoglobin and adult hemoglobin. Myoglobin and hemoglobin are both oxygen-binding proteins, but they serve different functions and we can find them in different locations within the body. Myoglobin is primarily located in muscle tissues. In the muscle tissues, it stores and releases oxygen to meet the metabolic demands of the muscle cells during periods of intense activity. Also, myoglobin has a high affinity for oxygen in comparison to the adult hemoglobin, and this allows for efficient extraction of oxygen from the hemoglobin of the blood. On the other hand, hemoglobin is found in red blood cells and is responsible for the transportation of oxygen from the lungs to the rest of the body, as well as returning carbon dioxide to the lungs for exhalation. The ability of hemoglobin to bind and release oxygen is regulated by pH as well as the carbon dioxide levels. So from this oxygen dissociation curve, we can see myoglobin has a higher percentage saturation of oxygen compared to the adult hemoglobin at all partial pressures of oxygen. And this ensures that myoglobin will always be able to take away oxygen from hemoglobin in order to ensure that the muscles receive oxygen required for aerobic respiration to produce ATP that is required during muscle contraction. So since ATP is required for muscle contraction, we can see the muscle fibers or the muscle cells have many mitochondria and they contain many myoglobin. The myoglobins are there to deliver oxygen and the mitochondria are there to carry out aerobic respiration, and this ensures that contractions can be attained in order to perform specific activities within the body. So the types of muscle fibers include the slow twitch muscle fibers as well as the fast twitch muscle fibers. Slow twitch muscle fibers are adapted for steady action over long periods of time. They contract slowly, but they can stay contracted for longer periods of time. They're used to maintain body posture as well as for long periods of activity, and they have plenty of myoglobin. They also have big networks of blood to deliver glucose as well as oxygen for aerobic respiration in order to carry out the contraction for longer periods of time. On the other hand, fast twitch muscle fibers are adapted for rapid as well as sudden activity. They contract rapidly, however they do fatigue quickly. These are usually used anaerobically. They have low levels of myoglobin and they have fewer mitochondria. They cannot produce high levels of ATP, of course, because there is anaerobic respiration. Less ATP is produced, so the muscle contraction cannot be maintained for longer periods of time. Fast twitch muscle fibers have more myofibrils packed within them, and they have a lot of glycogen stores. Slow twitch muscle fibers are usually used among marathoners, while these are used for sprinters. Going on to topic 7b4, how muscles contract. Here we are looking at a sarcomere, one that is relaxed and one that is contracted. In the contracted muscle, we can see the actin filaments have been pulled closer and the sarcomere is shorter in comparison to in the relaxed state. Arrival of an action potential at the neuromuscular junction triggers the release of calcium ions from the sarcoplasmic reticulum of a muscle cell. The calcium ions are going to bind to troponin, and this causes a conformational change that moves the tropomyosin away from the myosin binding site on actin filaments. This is the exposed site, and we can see the binding is going to occur. The myosin head then binds to the exposed site as ADP and PI are released from the head. Now, this resultant power stroke pulls the actin filaments towards the center of the sarcomere. ATP attaches to the head, and the head is detached from the binding site. ATP is going to be hydrolyzed into ADP and PI. The cycle of attachment, pivoting, detachment, and reattachment is going to be powered by hydrolysis of ATP, and it's going to continue as long as calcium ions and ATP are present, resulting in the shortening of the sarcomere and muscle contraction. This is what we can see. The muscle is contracted because the sarcomere has been shortened, or the many sarcomeres are shortened. When the muscle relaxes, calcium ions are pumped back into the sarcoplasmic reticulum. The tropomyosin covers the myosin binding site on actin, and the muscle fiber returns to its resting state. Going on to topic 7b5, the cardiac muscle and control of the heart. The cardiac muscles are myogenic, meaning they contract without external stimulation. They also have an intrinsic rhythm, which is built in. See, this is the origin of the electrical stimulation. We call that the sinoatrial node, and this ensures a faster intrinsic rhythm. This is the pacemaker region. Electrical stimulation is going to be released from this region, and then it's going to spread out to all the tissues of the heart. When depolarization occurs, there is going to be a momentary delay before excitation of the AVN, and this ensures that all the atria are contracted to ensure even depolarization. After atrial contraction, the electrical excitation is going to spread through the band of His to the Purkinje tissues. The Purkinje tissues consist of conducting fibers, and these penetrate through the septum, and they spread the electrical excitation throughout the whole ventricles or through all the ventricles, and this causes ventricular contraction. As the ventricles contract, they will squeeze the blood, pushing it away from the heart in order to go to the blood vessels leading away from the heart. So the condition of the heart can be studied using electrocardiography. Electrocardiography is a non-invasive diagnostic tool that records the electrical activity of the heart over time. It uses electrodes that are placed on the skin. We can see that here. This provides a visual representation of the heart's rhythm, the heart rate, electrical conduction pathways, and so on. And it's also used to detect abnormalities within the heart. These abnormalities could be arrhythmias as well as other heart conditions. In using the ECG, they can use ethanol to wipe the skin. This ensures any oily substances or things that will interfere with the electrical conduction are removed. It's carried out when the patient is lying down. And sometimes they could carry it out during exercise to see the heart's condition during exercise. The ECG trace consists of characteristic waves and intervals. We can see the P wave, the QRS complex, as well as the T wave. The P wave represents atrial depolarization. We can see when the atria are going to contract. This indicates the initiation of the heartbeat in the sinoatrial node. We can see this position here, and contraction of the atria. So this part represents the P wave. Depolarization of the atria as well as atrial contraction. The QRS complex signifies ventricular depolarization, triggering the contraction of the ventricles. So we can see spreading through the ventricles and causing the ventricles to contract. And then the T wave reflects ventricular repolarization. This is the recovery phase when the ventricles prepare for the next contraction. Here we see the electrical and pressure changes during the cardiac cycle. I've already talked about the P wave, the QRS complex, as well as the T wave. This is for atrial systole. This is for the depolarization of the ventricles resulting in ventricular contraction, and this is for the recovery or the repolarization of the ventricles. Here we can see the aortic valve begins to open when the ventricles are beginning to contract. The ventricles are decreasing in volume since the aortic valves have opened so that the blood leaves the ventricles. The pressure within the aorta is increasing, and that is because more blood is flowing into that, and the pressure is going to be increased because the ventricles pump blood through the aorta, and the blood is going to be at high pressure. After again we see the cycle repeating, the ventricles increasing in volume as the atrial pressure is decreasing because more blood is leaving the atria to go to the ventricles. Moving on to topic 7b6, the principles of homeostasis. Homeostasis is a process of maintaining internal dynamic equilibrium by responding to external and internal changes, which could be the stimuli. The responses can be to temperature, pH, water potential, salinity, and so on. And control can be both by the nervous system as well as by hormonal systems. During control, effector organs are going to be stimulated to cause a corrective change. Here we see in homeostasis, meaning the conditions are controlled, but there is going to be an imbalance. The receptors are going to detect the change that has occurred, and then they're going to send nerve impulses to the control center. The control center is going to respond by causing the effector to make a corrective change. Like I said already, communication can be by hormones where a chemical messenger is used, or it could be through nerves where electrical messages are used. The feedback systems correct the overshoot or undershoot in order to maintain dynamic equilibrium. Feedback systems include positive feedback and negative feedback. Positive feedback works by increasing the effect that has triggered the response, while negative feedback works by maintaining a condition, for example, concentration of something within a specific range. Here we can see examples of negative feedback. Remember, negative feedback works by maintaining a specific system within a range. For example, we have blood glucose. Like we said already, receptors are going to detect the change in the specific conditions. And then a control center is going to be notified. The control center will cause the effectors to make a corrective change. In this case, it's returning the system to equilibrium. So here we can see if the stimulus is that the blood glucose has raised, insulin is going to be produced. Insulin triggers the liver cells as well as the cells of the body to take up more glucose. The liver cells will convert glucose to glycogen, while cells like the muscle cells are going to store for use. And these work together to ensure that the blood glucose is decreased to return the system to normal. If the change is that the blood glucose levels have fallen down, a hormone which is called glucagon is going to be released. Glucagon is going to stimulate the liver, so that glycogen stored within the liver is converted into glucose, and this glucose is going to be released into the blood so that the blood glucose concentration increases, again maintaining a fine control. During positive feedback, these work by intensifying the change that has occurred. Examples include in fruit ripening, contraction during childbirth, and blood clotting. We see there is going to be intensification of the change that has occurred. For example, during childbirth, the baby pushes against the cervix and the cervix is going to be stretched. The stretch receptors in the cervix are going to detect the change and they will send impulses to the brain. This is to the pituitary gland. And then the pituitary gland is going to release oxytocin. This is the hormone that causes the intensification of the contraction of the uterine walls. So the oxytocin makes the uterus contract harder, pushing the baby against the cervix. In topic 7b7, controlling the heart and the breathing rates, we begin with response to demand and cardiac output. During exercise and periods of high activity, lactate is going to build up, and the intrinsic heart rhythm alone cannot cope with the changes that occur, so the heart has to beat faster and harder to get rid of the excess lactate. So cardiac output has to change in negative feedback control mechanisms. The heart responds by changing the cardiac output, and this includes changing the heart rate as well as the cardiac volume. You need to remember this formula: cardiac output is equal to cardiac volume, which is in decibels cubed, times the heart rate, which is beats per minute. So looking at control of the heart, this can be hormonal or it can be nervous control of the heart. In nervous control of the heart, this is controlled by the cardiovascular center in the medulla oblongata of the brain. Chemical as well as stretch receptors in the lining of the blood vessels and the chambers of the heart will send impulses to the cardiovascular control center. These changes are going to be controlled by the autonomous nervous system. This nervous system is broken down into the sympathetic nervous system, which is excitatory, and the parasympathetic system, which is inhibitory. So nerve impulses are sent down from the cardiac control center through the sympathetic nerve to the heart to release noradrenaline to stimulate the sinoatrial node, increasing the heart rate. Or they could be sent down through the parasympathetic nervous system to release acetylcholine, which causes inhibition of the sinoatrial node, slowing down the heart rate. We also have baroreceptors, mechanoreceptors, and chemoreceptors. For baroreceptors, these are found in the carotid arteries as well as the neck of the aorta. They are sensitive to pressure changes, and at rest, they send a steady stream of signal to the cardiovascular control center through the sensory neuron. When exercise begins, the blood vessels dilate. This is a response to adrenaline, and due to this dilation, the blood pressure is going to fall a little, and this reduces the stretch on the receptors. This change is going to be detected, impulses are sent to the brain, and then a signal is going to be sent down through the sympathetic nerve to stimulate the heart, increasing the blood pressure by constricting the blood vessels. And after exercise, the blood pressure increases and the heart continues to pump harder. The baroreceptors are going to be stretched. They then send impulses to the cardiovascular control center, which responds through the parasympathetic system, causing the blood vessels to be widened so that the pressure can be decreased. Then we have the chemoreceptors. These are found in the walls of the aorta as well as the carotid arteries. During exercise, the higher concentration of carbon dioxide decreases the pH of the blood, and this is going to be detected by the carotid and aortic chemoreceptors, sending an impulse through the sensory neuron to the cardiovascular control center in the medulla. Impulses are going to be sent down through the sympathetic nerve to the heart. This causes an increase of the heart rate. So carbon dioxide is going to be released, and this raises the blood pH. Also, if impulses from the chemoreceptors to the cardiovascular control center in the medulla decrease, the impulses sent back through the sympathetic nerve also decrease. The acceleration of the heart is going to be decreased as a result. Looking at hormonal control of the heart, at hormonal control, we're going to be looking at this part here, and how it involves the heart, because it's going to lead to the production of adrenaline, and adrenaline has effects similar to noradrenaline, causing stimulation of the sinoatrial node and increasing the heart rate. When somebody is stressed, or when they're excited, or when they're nervous, the sympathetic nerve is going to stimulate the adrenal medulla to release adrenaline. Adrenaline will move around the body and bind to receptors on target organs, including the sinoatrial node, and this increases the frequency of excitation from the pacemaker region, leading to a faster heartbeat to supply more oxygen and glucose for muscle contraction. Moving on to topic 7b8, the response of the breathing system to exercise. We begin by looking at the components of the lung volume. We know during exercise, the muscle cells will need more oxygen, but they're going to produce more carbon dioxide, so the lungs have to function better, or the intercostal muscles have to be triggered to function better in order to bring in more oxygen and get rid of the carbon dioxide. So here we're going to begin by looking at tidal volume. This is the volume of air that enters and leaves the lungs when you breathe in naturally. The inspiratory reserve volume, which we can see here, this is the volume of air that can be taken in above the normal amount of air that is inspired. This is basically when you make a deep breath. The expiratory reserve volume, which is this one here, this is the volume of air that can be forced out above the normal expiration. This is air you take out when you exhale as deeply as you can. Then we have the vital capacity, this one here. This is the total of the tidal volume and the expiratory as well as the inspiratory reserve volume. The total lung capacity, we see this one here. That is the sum of the vital capacity as well as the residual volume. The residual volume, which is this one here, is volume of air that is left in the lungs after the strongest expiration possible. Inspiratory capacity is the volume of air that can be inspired from the end of a normal expiration. It is the total of the tidal volume plus the inspiratory reserve volume. We got to control and regulation of breathing. So this is controlled by the ventilation center in the medulla, and it's involved in the feedback system based on the stretching of the bronchi during breathing. The ventilation center contains the inspiratory center that controls breathing in. There is also an expiratory center that controls forced exhalation. Impulses from the ventilation center are going to travel down the sympathetic nerves, causing the intercostal muscles and the diaphragm to contract, and this makes the person inhale. The lungs will inflate. Stretch receptors in the walls of the bronchi send impulses to the ventilation center. The more the stretch on the bronchi, the more impulses sent. Then the impulses finally inhibit the ventilation center. It then stops stimulating the breathing muscles. Breathing is reduced as the muscles relax. This is the summary about respiration and homeostasis. Once exercise begins, the brain cortex is going to consciously recognize the beginning of movement, and it's going to send impulses to the ventilation center in the medulla. Also, changes in pH due to carbon dioxide and oxygen variation are going to be detected by chemoreceptors in the aortic bodies and the carotid bodies. They send impulses through the sensory neuron to the brain, and impulses from the brain are sent to the intercostal muscles of the breathing system to cause contraction so that more carbon dioxide is removed and more oxygen is supplied. The chemosensors and stretch receptors in the muscles of the lungs work together to notify the respiratory center to maintain increased ventilation until exercise is completed. Here the summary about how the whole process is stimulated. We can see the sensors here, or the receptors here and here. They detect the changes, they send impulses to the brain. This is partly in the heart. Also, those in the lungs are going to send impulses, and together they work, creating a homeostatic control that maintains ventilation as well as the heart rate in dynamic equilibrium. So this brings us to the end of this video on muscles, movement, and the heart. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.